Today the topic of lecture is CRISPR. Actually, what CRISPR is? Basically, CRISPR is the abbreviation of clustered regularly interspaced short palindromic repeats. But what the CRISPR is? Before going to details, I will explain different concepts present in this word CRISPR. First one is palindromic repeats. Then I will explain interspaced short palindromic repeats, and at the end of the lecture I will explain what actually clustered regularly interspaced short palindromic repeats are. So what are palindromes? A palindrome is a sequence of characters or numbers that are read same forward as it does backward. There are different types of palindromes in the world. For example, world palindromes. These are the sequence of the letters that form palindrome. For example, rotator. If you read rotator uh, forward or backward, it will pronounce the same. Madam, civic. These all words are palindromes. The second palindrome is number or numeric palindromes. These are the sequence of digits that form palindrome. For example, you can see the numbers. If you read them forward or backward, they will be same. So the palindrome word is actually used in all the fields of sciences, but in molecular biology or genetics, why we use the word palindrome? So the palindrome word in genetics or molecular biology. So there is a word palindromic repeat are used in molecular biology. Basically, a specific repeated DNA sequence arrangement, which we can say tandem repeat or microsatellite. This is common and usual sequences which are being repeated again and again in the genome. So tandem repeats are the sequences of DNA where a short sequence of nucleotides is repeated. So these nucleotide sequences repeated multiple times consecutively in the genome. If this repeated sequence read the same forward and backward, it is considered palindromic repeats. If we say, for example, the nucleotide sequence to be considered as palindrome in the DNA, if the complementary strand is read as same but in opposite direction. For example, here we have one strand of the DNA 5 prime to 3 prime which have the sequence CGATCG. Now look at the opposite or complementary strand. You can see the same sequence but in opposite direction. If you are reading it from 3 prime to 5 prime, you will see GCTAGC. So these sequences are the palindromic repeats to each other. If you look at the picture, you can see different repeats. We call them short palindromic repeats. If you see in the red circles, you will see the palindromic repeats. But there is a space between them. Therefore, we use the word interspaced. But these sequences, these palindromic repeats, are short and having space in between them. Therefore, we say them interspaced short palindromic repeats. And the palindromic repeat, which is repeating again and again, is GAATTC, which is from 5 prime to 3 prime, and the opposite or complementary strand is from 3 prime to 5 prime. Now it is clear that interspace short palindromic repeats are present in the genome. So these palindromic repeats are happen in the genome in a regular manner, in a regular way. Therefore, we call them as clustered regularly interspaced short palindromic repeats. So the CRISPR is actually a revolutionary technology in a molecular biology and genetics, and in this system which allows the scientist to make precise changes to an organism. So what kind of changes to an organism can be done by the scientist? It can be added, several nucleotides can be added, they can be deleted or can be replaced. So these three different type of changes can be done by the scientist through a technology which is called CRISPR. We will continue the lecture further. Thank you so much.